All right, fantastic. So yeah, real pleasure to have today's speaker as Moataz Assem from the University of Cambridge at the MRC CBU. So Moataz trained in, he's originally from Egypt. He trained in medicine at Alexandria University, then undertook training in biomedical engineering in Turkey and at MIT in the States. And then he has done his PhD with John Duncan at the MRC CBU in Cambridge. And I got to know or heard about Moataz since he gave his talk in receipt of his EPS prize for his PhD work at the EPS meeting recently. And he's been doing some fantastic work with John on higher cognition and the notion of domain general processing, particularly in using human brain imaging. So we're really looking forward to your talk, Moataz, and thanks so much for coming to tell us about everything you've been working on. So take it away, over to you.
Hey, thank you so much, Steve. And thanks again for having me. I hope you will enjoy this. I can also see Chris Frith, so thanks for initiating the prize, Chris.
Yes, I should have said it was the Frith prize, right? So,
yes.
Yes, there we go.
Okay, I'll get started. So I'll mainly be presenting work that I've done during my PhD, but also there are some new extensions from I'm now just starting my second year in postdoc at the CBU, and there are some new results that I've also just gotten yesterday. So hopefully you'll find it a bit exciting. Okay, so I'll start by, let me change. Can you see my mouse, by the way? Yeah, okay, great. So I'll start by giving a bit of a theoretical framework guiding us to look into the brain, and then I'll dive into what brain regions we're interested in and how we think about how they work. So the main theoretical question driving us is there core function behind Spearman's G? You might have heard about Spearman's G or general intelligence as the factor which describes how lots of tasks, performance on lots of tasks positively correlates with each other. In other words, there are some people who tend to perform really well on lots of different tasks, basically. And we are wondering whether there is really core cognitive function behind this Spearman's G. There is one specific type of task which correlates really strongly with G, which is usually these are like novel problem solvings and like these Raven progressive matrices. So in this example, for example, the participants were asked to try and solve this Raven matrix by drawing their answers in the box below. So if I give you enough time, maybe you'll figure out that the answer is something like this. And if you correlate the proportion of correct answers for these participants with their IQ scores measured independently, then you will see here, as shown in the blue dots, how indeed performance on such a task correlates positively with their IQ, such that people with higher IQ perform better on such a task. But let's, what about people who struggled? Let's delve deeper into a little bit what's involved in solving this task. So there was another modified condition for people to solve where the task was broken into steps for them. So they first just had to solve where should the vertical line be present. So hopefully you can see that the vertical line was in the middle. Now it's to the right. So here it's to the right. So the solution should be that the vertical line should be in the middle. And then they were asked, where should the arrow be pointing? So hopefully you can figure out that the arrow was pointing right. Now it's pointing left. So here it's pointing right, should be pointing left now. And the last step was that the left end stayed the same, so it should also stay the same. So when you give the same participants these broken down problems, they tend to perform pretty well regardless of their IQ score, basically. So what this manipulation pointed out is that perhaps there is something really important about breaking down a task, a complex task, into its simpler steps so that the participants can be able to solve it. So let's look at what it means to break down a task into simpler steps using a more everyday task, for example. So let's say you want to travel to Japan, and then you ask your question, what should I do with my right hand? Then you wouldn't be able to answer that question because there's a big gap between that very top goal of traveling to Japan and what to do with your right hand. But if you try to break down the task into simpler steps, okay, to travel to Japan, I need to fly, which means I need to buy a plane ticket, which means I need to access the web, and then I need to use the mouse to start typing on in the search box, for example. And then you're able to say, okay, what I'll do with my right hand is to move the mouse. So you need this sort of hierarchical representation of the tasks where also each step becomes simpler and simpler to be able to solve it. And this is on the right here, this is an illustration of how a problem can be broken into its simpler steps, the same problem here of traveling to Japan. And an important bit of breaking a problem or segmenting it into different steps is that also each step means you're linking the right parts related to it in the right structure and the right components, right? So step number one, you need to integrate the goal of traveling, perhaps the memory that Japan is far away, with the sub goal of flying, and then you move down the problem. And then the last step, for example, here would be that you have a goal, which is to type something on the screen. With the memory of needing to move the mouse, you move the mouse and linking it with the action of moving your hands. So if we look at what is involved in each step here, each step is basically a computational structure, or as we call it, a cognitive structure, linking the relevant parts from the different ingredients present in all around in the brain, linking them in the right structure and the right pathways. So this is, for example, how cognitive structure for step number one would look like. And this is how a cognitive structure for the last step, step N, for example, would look like. So this is basically the broad picture or the broad lens through which we look at how higher cognitive functions look like. And I think it can apply to all kinds of cognitive functions if we sit down and think carefully about it, where we need to build these sort of cognitive structures for every step as we move in solving a problem. So now I'll move into the brain bit. So how do we look into the brain for such a system that assembles a cognitive structure and assembles it in a flexible way? There are lots of ways to look at that. I'll mainly here be focusing on using functional MRI in humans, but I've also had some. We have an experiment where we used ECOG, for example, and show that there are some ways to look at it also in electrophysiologically. But I'm not sure if there will be time to cover that. But happy to talk about it after the talk as well. So with fMRI, so with fMRI, a conventional way of trying to find where this cognitive structure assembler is to contrast a task with a baseline, and this baseline could be anything. But what our lab has been traditionally using is to contrast the same task a harder version of the same task with an easier version of that task, because that would be the most controlled aspect. And the logic here is that when you're doing the harder task, that cognitive structure assembler is linking more and more parts with each other, more than the easier one. So with the logic of cognitive subtraction, you would end up with the system that is involved in linking the relevant parts of that task. And then if that cognitive structure assembler is really domain general, then it should also be co-activated across lots of different tasks. So I'm here highlighting one really nice study by Evelina Fedorenko in 2013, where they looked at the average of 7 and the conjunction of 7 hard versus easy tasks. So on the screen here, it's the average, but in the study, they looked at the conjunction of single voxels, and they indeed found that there are single voxels in the brain that show overlaps across these 7 tasks. And these were ranging from just looking at some non-words versus words, to doing some response inhibition tasks, to doing some math tasks, to doing spatial and verbal working memory, lots of different types of tasks. And as you can see, as expected, there is a system that seems to be present and involved in each of these tasks. Some has components in both the lateral, frontal and parietal regions, and also components in the medial, frontal and parietal region. So what about using intelligence tasks like the Raven matrices I presented to you in the first slide? So using the CAN data, which is a big aging data set, we had about more than 250 subjects that solved, again, a hard versus easy fluid intelligence task. And hopefully you can see that almost the same regions again in the lateral frontal lobe, in the lateral parietal, and in the medial surfaces are present. But obviously you can see that there are also some differences, and these differences are going to be important, and I'll highlight them later in the talk. And we also know that if people get lesions in these regions, I forgot to mention that, of course, if you've done any fMRI study, you would have seen these sort of activations at some point, and they come in different names: the task positive, cognitive control, frontoparietal activations. We like to call them multiple demand system. That's the name my supervisor John Duncan coined about more than a decade ago, mainly because it's just the simplest description that these are regions that are activated by multiple demands, multiple cognitive demands. So we know that, and I'll be mentioning using the term MD just as a short for multiple demand system. So we know that these MD regions, if they are damaged, and if the lesion volume damages more and more of them, then people start dropping in their IQ scores. But if you use a control region in the brain, for example, the language network, then this has no effect on their IQ score. So this tells us that there are indeed very specific regions distributed across the brain that seem to be important in cognitive control and solving complex problems, basically. But we've known this for many years, even since the early days of doing fMRI. But there's still no precise consensus on where these regions are really present. We roughly know some of them are like in the middle frontal gyrus, some of them are in the anterior cingulate cortex, but we also know that using these terms and using curvature is not a reliable way of labeling functional areas in the brain. So we really didn't have a good idea of where exactly they are present. And we also, there were criticisms about the idea of functional preferences. So hardcore anatomists will tell you that these regions, every patch in the brain has a specific function. The regions in the brain are specialized. They have unique connections to each other. So this presents an important challenge to the idea that there are regions in the brain which are doing almost everything, that they are domain general in function, basically. And we also don't know much about their connectivity profile, whether at an anatomical level or even at a functional level. we think they might be well connected with each other, but we didn't really know how to relate them with, for example, these canonical resting state networks like the frontal-parietal networks, the default mode networks. Like, is there some overlap between them or not? So when I started my PhD, we realized that with the current imaging techniques are not going to be able to help us resolve these questions in a good way. So we turned into the Human Connectome Project imaging approach. And what this approach offers is much higher spatial resolution in finding where these activations are in the brain, about approximately 3 times better than the traditional imaging ways. And this is for lots of reasons. They've developed these pipelines for many years, but there are 2 really important aspects here. First is that they respect the cortical geometry, so they really use surface-based approaches. And the second is that they align the cortices across subjects using multimodal criteria. So traditionally, if you align your images to M&I space, you're basically depending on a cortical curvature. But we know that cortical curvature is not consistent even across twins, and especially in the higher association areas. So what the HCP team here has done is to align the cortices together using more consistent features like myelin maps, like connectivity maps, for example. And this has really improved the results, as you will hopefully see shortly. What this approach also offers us is that we can interpret the activations across or against a neurobiologically motivated parcellation. So using hundreds of data from hundreds of subjects, the HCP team was able to divide the cortex into 180 regions per hemisphere. And each border was defined using shifts in multimodal criteria. So for example, that the myelin content was shifting from high to low, that the connectivity was shifting between one area to another. And this provides a really good way of relating your activations to a common template that is also neurobiologically motivated, and not just each lab using their own functional ROI, and then all of the labs start to argue which ROI is related to which region of the brain. Yeah, so this is what we've done using the HCP approach. First, in our first experiment, we decided to use the HCP data itself. They had, we used data from more than 400 subjects, and we tried to, each subject did 7 tasks. We selected the tasks that were most relevant to our cognitive, the cognitive functions we're interested in. So they had a hard versus easy working memory task, which was an n-back task, 2-back versus 0-back. They had a hard versus easy reasoning task, which was trying to relate or say whether different features of abstract shapes were related or not. And they had a math versus story task. This wasn't an ideal task because it wasn't really like a hard versus easy math, but we know from lots of previous studies that math activates these same multiple demand regions. These the first two contrasts were visually presented. The math contrast was auditorily presented. So here I'm presenting the average activations of these 400 plus subjects, and hopefully you can see that there are 9 clear patches across the cortex that are quite separated and well delineated from each other that we didn't really see in previous studies, that sort of delineation from each other. So there is a dorsal frontal patch. There are 3 patches here along more ventral. There is a 5th patch here in the insula. There is a temporal patch, which is much more anterior than what we previously thought it would be. There's a lateral parietal patch around the intraparietal sulcus and its surrounding regions. And there are 2 medial patches here, one in the prefrontal cortex and one in the parietal cortex. Then we asked, how are these patches related to these HCP 180 parcellation scheme? So we did a conjunction for each of these contrasts. And what we found is that these patches are related to 28 regions out of the 180 regions from the HCP parcellation. Then we asked the question, we're like, okay, are there regions that are more activated than others within these 27 regions? And indeed, we found that within these 28 regions, there were 10 regions that were more consistently and strongly activated across each of the three contrasts. And we define that by more activated than the average of these 28 parcellations for 28 regions for each contrast. So we labeled those as the core MD regions and the surrounding ones as the penumbra. So you might think, okay, maybe this is an arbitrary division of core versus penumbra. Is there another way to support our division of that sort of division? And we looked, each subject had 1 hour of resting state data. So we looked at whether these core regions were more connected with each other. And that's indeed what we could find here in the first bar, that the core regions are much, much more connected with each other than their connections to the penumbra and then the connections to the other brain regions and the average connection of all other brain regions. So what this tells us is that there is a system that is involved in diverse cognitive tasks that is tightly interconnected with each other. And we can already say that this system is divided into at least two big portions, a core and a penumbra. So is this system limited just to the cortex? No, this is the only slide I have on subcortex, but so we looked at whether there's a conjunction in the subcortex and cerebellum, and indeed we find hotspots in the caudate, mainly in the head, but also extends to the body of the caudate, and also in the cerebellum, where most of the hotspots were present around crosses 1 and 2. And we also looked at this using resting state data. So we asked which of these voxels is more connected to the core MD regions in the cortex more than the surrounding cortical regions. And indeed, we replicated the caudate hotspots, but we also found an anterior thalamic region which wasn't present in the task contrast. Maybe because the SNR of the subcortex and the HCP sequences isn't that good. So this needs to be followed up using maybe a better sequences or a 7T study. But we also replicated the hotspots in the cerebellum around crosses 1 and 2. And this also confirms the importance of our definition of 10 core regions in the cortex, because these are just, we replicated these results by their connection to just those 10 regions and not more than, and not the whole cortex or the whole association cortices. So back to the cortex, what about the idea of functional preferences or functional specializations? So here I colored each parcel or each region with its preference to which task it was more strongly activated in. So more reddish colors means it was more strongly activated in the working memory task, more bluish colors means it was more activated in the math task, and more greenish colors means it was more activated in the relational reasoning task. And as you can see, there's a mosaic of colors here. And by definition, just to remind you, each of these regions was already strongly activated in each of these tasks. So what we're seeing here is relative functional preferences, not absolute functional specializations. And the only reason we're able to assert this claim is the sheer number of subjects we had. So remember, we had 400 plus subjects. So the statistical power we had was enormous. And if we split those subjects into 2 groups, 200 plus groups, then every little dip and change and preference in each of these regions was replicated almost identically by that other subject, basically. So these differences are real, but they are not absolute functional specializations. They are relative functional specializations. Just that previous studies had lower statistical power, so they were not able to see that it's relative rather than absolute. So this led us to develop this sort of a framework to try and understand these results or make sense of these activations, basically. And here, each MD region is schematically has a different color. The idea is that how can we explain relative functional preferences with also the co-activation paradigm? And the hypothesis we're proposing is that each MD region has access to a specific type of information, some related to its also local connectivity. So it only has access to some ingredients that are relevant to the task. So this is where the relative preferences will emerge. But because these MD regions are strongly connected with each other and they probably communicate quite quickly with each other, then you also get this sort of co-activation pattern where the frontal and parietal, lateral and medial parts co-activate with each other. So we wanted then to test this hypothesis with also more tasks, because these results from the ACP dataset were just 3 tasks, right? So to remind you, this is how the 9 patches looked like using 400 plus subjects. And then we decided to do a new experiment, collecting data from 4 more tasks at the CBU, using the HCP sequences, analyzing it using the same approach. And these were the 9 patches from the 4 new tasks that we used. And they look quite similar. We were very pleased. But also note that we just replicated these 9 patches using a much smaller number of subjects. So this tells you, hopefully shows you the quality of these HCP sequences. And the 4 tasks we used an N-back task again, a working memory task. This time 3 back versus 1 back. This was like a sanity check for us to compare it with the HCP N-back activations to know that the sequences have worked well with in our department as well. We had a switch task and we had a stop signal task. So all of these tasks were block designed. The N-back was 3 back blocks versus 1 back block, so hard versus easy blocks. The switch ones, they were blocks where the participants had to switch between solving with 2 rules versus solving with 1 rule only. And the stop signal one was blocks where the participants stop blocks where there's a stop signal that appears versus a block which didn't have any stop signal. And the N-back task had an auditory and a visual version as well. So this is how the average looks like, but we know group average maps can be misleading. So what about like conjunctions of these tasks at the single subject level? So this is exactly what we did. We asked which vertices were significantly activated in each of these 4 tasks across within a single subject. And then this is a probabilistic map of their activations across subjects. And hopefully you can see that the regions that overlap quite nicely. The highest overlaps overlap quite nicely with the MD borders, which are colored here in blue regions, basically. But you can also note that some regions are a little bit weaker than others, and some are probably non-existent, like this temporal patch, which is again going to be important in the next few slides. But overall, this picture tells us that, again, there is a common sort of system involved in these 4 tasks. And hopefully you've noticed that these tasks were targeting the executive function domain, the unity and diversity claims. So this is what we are what we relate to the unity part of the executive function tasks. What about the diversity bit of the executive functions or the relative functional preferences? So here I colored each vertex, so not just each region, each vertex with this preference for each of these 3 tasks, basically. And you can see that there's again a mosaic of colors present all around the brain, and there's a lot to unpack here. And this is what I'm going to do next in the next few slides. But before I do that, I want to make you believe that every little color change here, which are just a few millimeters apart, is real and and needs to be interpreted in a neurobiological way. So this is how I'll hopefully convince you with it. So remember the NBAC task, we had a visual and an auditory version. They were done on 2 different sessions, session 1 and session 2. The same subjects did the same task. And this is the contrast for the 3-back versus 1-back, so hard versus easy. One of them is visual and one of them is auditory. Can you tell which one is visual working memory and which one is auditory working memory? Any guesses?
Session 2, auditory.
Yeah. What gave it away?
Just, I mean, it's a slightly redder temporal lobe, but that's about it.
Yeah. But what about like front parietal activations? It's almost identical across both of them, right? Yeah. So this sort of similarity, it was non-existent before we had the sort of HCP sequences, right? And this is not just at the group level, this is also at the single subject level. So single subjects were quite almost identical to each other in activations. So what I want you to take away from this slide is that every little shift in activity is going to be meaningful, and it's something that we really need to understand. Why did it shift, and try to interpret these sort of shifts, basically. So now back to the 3 tasks that we had. Here I'm focusing on just one region in the lateral prefrontal cortex called P946B. And I colored here the vertices in each region with, again, its preference for which task it was more activated in. So more bluish vertices are stop, more reddish are NBAC, and more greenish are the switch task. And then I placed seeds using independent resting state data. So resting state data that is not related to in any way to our current data. And I looked at how these seed connectivity patterns look like. So this is the map for how a seed in location number 1 looks like. And hopefully you can see that this seed is more connected to the regions near the border, along this black border, both in the frontal and the parietal regions. And this is how seed number 2 connectivity map looks like. So hopefully you can see also that now the activations have started to shift slightly more posteriorly, both in frontal and parietal regions. And this is seed number 3. So hopefully you can again see that the connectivity shifted more posterior. So now this seed is more connected to regions along that more ventral border and more connected to more posterior parietal regions. This tells you that even within a single MD region, there are some fine-grained preferences at the resting state level, right? So now compare that to the activations of each task separately. So hopefully you can see that seed number 1 connectivity map looks quite similar to the task activations from the stop task. So again, the stop task has activations here exactly at that black border, both in the frontal and the parietal regions. And hopefully you can see also seed number 2 is more similar to the NBAC activity, which has shifted a bit more dorsal and posteriorly, both frontal and parietal regions. And seed number 3 has activations which are similar to the switch one. Sorry, has connectivity similar to the switch activations, which are more shifted towards this more ventral border here, and also its activity is stronger in the posterior parietal regions. So what this tells us is that these sort of relative functional preferences that we see in tasks must be related somehow to these sort of fine-grained resting state architecture here. And what I'll show you in the next slide is basically quantifying this in a more automatic way rather than me just manually placing seeds here. So the question I asked is whether seed number 1 is more connected to task 1 or task 2 or task 3, and then after also excluding all of the connectivity of the vertices that surround it, both on the same hemisphere and the opposite hemisphere, because we know that hemispheric connections, we know that local connectivity of the connectivity of the vertices near each other can be similar just because of the smoothness of the fMRI signal. And this is how the map looks like. So hopefully, again, you can see that these regions shift in a specific topography, similar to what I've shown you. So stop and back switch, and then you have stop and back switch. So it's going like this here, and it's going like this in the parietal regions as well. So how are these sort of shifts or resting state fine-grained patterns relate to what we know about canonical networks? So here I've colored the resting state networks that were produced from another independent set of HCP data by the Cole et al. Lab in the US. And I tried to color the resting state networks in the same way. So hopefully you can see that it's more fine-grained in the top map, but hopefully you can see that the bluish regions are around the same area, the greenish regions are around the same areas here as well, and the reddish regions are around the same area here. So bluish would be single opercular, green would be dorsal attention, red would be the fold mode network, and the yellow would be the frontal parietal network. So what this tells us that at a coarser level, these sort of shifts are also related to where the canonical resting state networks are located, basically. So what about task activations? So if we try to quantify the activations of each canonical resting state network, so the frontal parietal network here was split into the core MD regions, which form a subset of the frontal parietal network, and the non-core MD regions. And as you can see, more or less the activations here for each of the tasks are quite equal to each other, except, for example, you see a preference of the stop signal for the right hemisphere and the switch signal, and the switch task for the left hemisphere. But what about, it gets more interesting when you look at these other 3 networks. So the dorsal attention seems to have a preference for the switch task. The single opercular seems to have a preference for the stop signal task, and the DMN seems to be the least deactivated by the working memory task. And all of this survives statistical comparisons and correcting for multiple comparisons as well. So what this tells us is that it seems that each cognitive task activation is a mixture between an MD activity and a more specialized network. And this is what hopefully the stop signal contrast is the one that shows this the most, basically. So here I colored the MD network with the green borders, and I colored the single opercular network with the blue borders. And hopefully you can see that the activations lie exactly at the border. You know, it's in between these 2 networks, basically. So this tells us that stop signal has something to do with the single opercular network, which has also some different names in the literature, like the salience network. And it could be that the salience element of the stop signal is what's driving this connection with the MD network. But we also found it quite interesting that the activations were lying quite exactly at the border. So it was, and remember, these borders aren't just defined by one criteria, they are defined by multimodal neurobiologically motivated criteria, like the myelin content, the cortical thickness, and in some regions, the curvature as well. So we're wondering whether this pattern highlights something special about the borders. So we asked the question of which regions in every single subject and in every task had the strongest activations. And then we created an overlap map for these strongest activations. And as you can see here, every colored vertex in red here means it survived that threshold, and the bright red ones means that it lies at a border. And if you quantify these borders, these vertices that are bright red, you see that most of them actually lie at the borders of MD regions, which are colored in blue here. So what this tells you is that it seems that the strongest activations lie for each of these tasks lie at the borders of MD regions. They don't just lie within the MD regions. So there's something special about these borders here. And you might ask, okay, is it just any border, any MD border, or is it related to also the borders of the rest of the MD region with its nearby regions? And here I colored each vertex with its preference for each task, so that if that vertex had more overlapping subjects for one task, it took the label of that task. And hopefully you can see that the activations peak not just at any border, but it peaks at these task relevant borders. So I tried to highlight some of them here. So these are the task preferences at the group level. Green is switch, for example. So you can see that the green vertices that lie at the border are in the same location where the switch task has a stronger preference. The bluish vertices here also are where the stop signal task has a preference, and the end-back task here has the end-back vertices also lie around the end-back task preferences here in red as well. So this is quite new, because this is a new level of looking at brain anatomy in relation to task activations, and we're not quite sure why exactly the activations are at the border, but we think that the borders must be related in an important way to the connections and communication between these more specialized regions and the more domain general brain regions. And this led us to develop this new hypothesis, which guided our, which is guiding our new experiments. So here is a blown-up picture of the intraparietal sulcus. So the yellow regions are the MD regions, the core MD regions here. And each of the other regions is colored according to its assignment in the resting state network. So greenish ones are dorsal attention, purple is cinguloopercular, bluish is language, DMN is more red. And we thought, okay, if we bombard these MD regions with lots of different tasks, then we should see these sort of patterns, that if you do a more spatial task, it comes from more dorsal, a more dorsal direction, and it should, the activation should somehow peak around these borders. If we do a more like salience task, it should be coming from that direction. A more languagey task should be coming from that direction, and a more like theory of mind or episodic memory recall task, then it should be coming from that direction. So this is where my EPS lecture stopped, but yesterday just got some new results from we did that experiment basically. Gavin, who's a PhD student I'm co-supervising with John, just sent me some exciting results. It's still preliminary, but I really am very excited about it. So I'll just show you quickly what we found. So in the language task, I colored I colored the language network in green here, and the core MD networks are in white. And this is the language hard versus easy. So hopefully you can see that the language activations also again peak. So they show a mixture of course, activating the language network, but also overlapping with the exact borders with the MD regions here. And the language task was verb generation versus noun repetition, and again, block design. And then we had a mental rotation task, and here the borders, the green ones, highlight the dorsal attention network, And hopefully you can see that now the direction of activation has shifted completely. So it's coming from the dorsal direction. And again, you can see some activations nearer to the borders with MD regions here and here as well. Of course, we have to quantify this. This is still very preliminary. We had a memory task, which was recollection versus familiarity. And here the red one, the red borders highlight DMN. So hopefully you can see that if you know the literature of recollection, then you see activations peaking around the TPJ, and very interestingly, it seems to be peaking around the exact borders of with MD regions here. And there is an MD region here, but it's not part of the core, but this is the border where it lies with. And you can see here that the memory activations are just activating, again, the PCC, right? The posterior cingulate cortex, where you expect memory activations with the MD regions, with an MD region here. And then this is a theory of mind task. So here again, I highlighted the DMN regions in green, and also an extra network, which is defined here and overlaps with the theory of mind activations in the HCP tasks. And you can see here that now the activations are approaching MD regions from that direction, from that sort of angle, which is very different, for example, from the angle where the DMN activations are approaching the MD regions here. I'm not sure if I managed to explain this well, but I literally just added these slides an hour ago. Yeah, this is basically, we're really excited about this. Still don't have the activations from the salience task, but we'll see how it looks like next. Okay, so before I finish, I just wanted to acknowledge the HCP team members we've worked with, especially Matt Lasser and David Van Essen, which were our collaborators in the first study because we used the HCP data. But also they continue to give us guidance in using the HCP sequences at the CBU. And of course, at the CBU, John Duncan, my PhD supervisor, Sneha, who we collected the executive function data together. We took over the CBU scanner for 3 months to just acquire 100 sessions from these subjects, because in the HCP sequences, they're longer and you need 2 sessions. You need at least 1 hour just to acquire the HCP requirements plus the task data you need. And Gavin Shields, who's the PhD student I'm co-supervising with John, who just I presented his new results, and of course the MRI methods and IT teams at CBU, because setting up these sequences wasn't an easy task. Of course, my funding from the Cambridge Trust during my PhD. So to summarize what I've just told you, this is a flat map of the MD patches I showed you. So there is a tightly connected brain-wide system. It's not just in the cortex. Remember, it's also in the subcortex and the cerebellum. That seems to be domain general. It's activated across multiple diverse cognitive tasks, but also, like I showed you, it shows relative functional preferences. Cognitive task activations seem to be a mixture of activating a domain general region plus a surrounding or specialized regions. And these sort of preferences seem to shift in ways which are which are not random, which have some pattern to it. So related to the fine-grained resting state architecture. And the activations also seem to be peaking at the borders, and especially specifically not just any border, but the borders between domain general and the domain specific regions. So this suggests a critical role for these borders in integrating information. And I'm done. Did I take too much time?
No, that's perfect. Thanks so much, Moataz. So we should first unmute and give you a round of applause for a great presentation. That was really great. So I'm just going to stop the recording and